And again, I'll be reporting on the rose rosette disease and rose, I guess, black spot disease resistance discovery work that we're doing here at Texas A&M. I, Dave, and Trish Klein work on this particular, I guess, area here at Texas. Our main focus is black spot and cercospora, and of course, rose rosette disease would be the work that is being done in Tennessee and Oklahoma, where the same materials that we work on are planted there as well. We were tasked with developing, I guess, the populations, linkage maps, diversity panels, and what have you, to then look at the diseases we can look at, and we focus on black spot and cercospora, and then Oklahoma and Tennessee will be focusing on rose rosette disease. And hopefully all these things will come together in the end to give us a good picture about the level of resistance, the types of resistances, and where these factors are in the genome. Our approach to using the information we're producing from this project is basically to understand better the genetic basis of these traits. And of course, we've got biparental mapping populations as well as diversity panels to get at that. We are part of another SCRI project, Tools for Polyploids, that is coming to an end. But through that project, we've developed and used a number of computational tools that allow genetic analysis of polyploids. And polyploid data is more complex than diploid data. So we now have the tools to be able to do the genetic analysis that we like to do for this type of project, and try to then take that and move that platform forward to do more breeding activities, for example. And one of the things that we're getting into is, of course, the use of genomic prediction methodology to try to get at a method to manipulate various sources of resistance simultaneously as we try to develop materials with better resistance down the line. This is the complex slide that I often show to show you the pipeline that we now have for genetic analysis in roses. And as I said before, this is partly from the Tools for Polyploids project, and Christina Gucci and Jikan Lau used to be postdocs in the program, which now have moved on to greener pastures. Chris is with Breeding Insight, a USDA project in Cornell, and Jikan Lau is now a USDA RS employee working on peaches in Georgia, I believe. And what you see here is a pipeline of tools that were developed through the Tools for Polyploids, and Chris and Jikan work to try to put those to work for this particular project. And the things that the two areas of activity that we're focused on is, of course, discovering new genes for resistance in the QTL gene discovery module, and of course, applying some of the same data for genomic prediction approaches in the genomic prediction module. And of course, on the phenotyping side, we've got a network of biparental populations with modern Fargo and modern blush as being the perceived resistance, I guess, genotypes for RRD. And of course, the other parents having varying levels of black spot resistance as well, in addition to a diversity panel that has, again, a collection of genotypes and a collection of alleles that we want to test to see if they also provide some level of resistance to black spot, in particular in our environment. So again, rose rosette disease is what they will see in Oklahoma and Tennessee. Black spot is our major focus here, in addition to cercospora, leaf spot, and other traits. And again, the sample sizes are about 800 in the case of biparental populations, about 400 in the diversity panel, more or less. Dave gave you the most accurate numbers, but this is just for illustration purposes here. And again, the traits, rose rosette disease, black spot resistance, and then some ornamental traits as well. With respect to phenotyping, Tessa Hockhaus, Navdeep Kaur are two PhD students, and Ivan Perez is a master's student that are working on the project. Since Chris and Jikan left, Tessa has taken on the leadership to handle the biocomputational work for this project, and has been training Navdeep and Ivan to do the same, who now have picked up the mantle appropriately. All three get together to do the phenotyping for this project. And what I show you here are the various biparental populations and a collection of varieties and breeding lines that we have in the field. It's about 2,000 to 3,000 lines or rose bushes out there that are phenotyped monthly for Cercospora leaf spot, black spot disease, defoliation, flower intensity, and flower number. And again, all of this within the context of the Sustainable Roses Project. And we'll keep phenotyping this material every year, monthly, to get a better feel for the level of disease resistance that these materials have. Another thing that we are tasked to do is to actually genotype the materials that we're testing phenotypically. And what I have here is a summary of what has been genotyped so far using the Rose Axiom Array. And that array allows querying 70,000 SNPs in the genome. And what we have here is a total of about 735 lines that have been genotyped so far. The first 3 rows show 3 new populations that we're trying to, I guess, focus on, in addition to minor ones that also have modern blush as a parent or modern fire glow as a parent. And these may be also members of populations previously analyzed by Stan Hokanson and David Lizak in Minnesota and Wisconsin, respectively. And what we've done is to try to augment some of those populations by genotyping those that hadn't been genotyped before. So in the end, we'll have close to 10 to 14 populations that have modern blush or modern fire glow in their background that we will be able to then analyze in conjunction to the phenotypes to get a better feel for the level of resistance and where these factors are and which ones are the most important ones, etc. These are, again, this is work by Navdeep Kaur and Ivan Perez in collaboration with Tessa, who's handled the DNA extractions as well and the genotyping, and has taught Navdeep and Ivan to run some scripts in R to build linkage maps of our 2 new large populations, which is Modern Blush by Miracle on the Hudson and Modern Fire Glow by Miracle on the Hudson in the bottom. And again, one population has about 198 individuals, the other one 172. And we've got 6,000 loci on one map and 4,000 loci in another one. And we've started to do some QTL mapping for disease resistance and ornamental traits. And so far, preliminary results are very, very encouraging. And we look forward to continuing to do that. So we've genotyped some material and we've started to do some QTL analysis. In addition to that, Jikan Lau, before he left, helped me work with Thermo Fisher Scientific to develop what we call a genotyping by sequencing solution, or targeted sequencing in this case, for roses. And what he did, Jikan did, was to look for about 1,500 markers in the genome. And then we worked with Thermo Fisher Scientific to develop a targeted amplification sequencing protocol for those loci. And we now have a panel of about 1,536 markers that can be, I guess, queried with this new sequencing technology. And Tessa has been instrumental in getting the DNA extraction, and she's in the process of looking at the data and seeing the outcome. And just for your reference, it cost us close to about $16,000 to create a marker set for 6,000 reactions. So this would be enough materials to do 6,000, to assay 6,000 lines and genotype 6,000 materials with about 1,500 markers. And that is to contrast with what we currently have, which is the Axiom Array that queries 35,000 SNPs, mostly, that are useful. And the cost there is $8,000. And this new panel, the cost to sample 96 individuals will be $1,200. So one of the things that we're trying to do right now is to validate how useful this targeted sequencing panel with Fisher Scientific actually is. But we look forward to having an alternative to the Axiom Array, because it's a bit more cost prohibitive compared to what this panel would be. Another interesting about this project is that once we validated the usefulness of this genotyping approach, then there would be a publicly available resource to genotype roses using this particular platform. and it would be basically $1,000 per 96 individuals at that point. All of the other costs of design and development of the panel have been made. So basically, we would have a public resource for genotyping for anybody interested down the line. Our plan would be to hopefully go up to 5,000 markers rather than just staying at 1,500 at this point. So again, this has been some work by Jikan and Tessa that I think has some important, I guess, repercussions, because we want to have more cheaper, I guess, genotyping for the community if possible. $8,000 per 96 samples is just very expensive right now. Just want to do a couple of introductions here. You've already heard about Natalie, who's helping us with project coordination, but I do have a new postdoc coming on board. His name is Rezwan Tariq, and you probably heard him asking questions earlier. He is native from Pakistan. He's been working at the King Abdullah University of Science and Technology in Saudi Arabia. He was working with Jesse Poland, who leads the plant science program there, and he's worked on the assembly of the purple flesh dragon fruit, and we look forward to having him come on board early next year as we are working through visa issues right now. And I think that's all I'll have for you. Again, thank you for USDA funding for this, the NIFA, and of course the Specialty Crop Research Initiative, both the Tools for Polyploids and Sustainable Roses that help us kind of tackle this issue. And again, thank you to all of our collaborators from industry as well. And I'm going to stop there. And if you have any questions, please let me know.
Oscar?
Go ahead.
Sorry.
Hi, Stan. How are you?
Turn mine on. Oscar, can you hear me? Oops. We're having some feedback here. Anyways, I wonder if you are planning to do fee for service with this new system you guys are developing there.
So the plan that we had with this targeted genotyping was, well, there's a number of service providers that provide that service. And we've had a relationship with Thermo Fisher Scientific because we use the Axiom Array, the Rose Axiom Array through them. And they basically came and said, well, why don't you look into this other alternative, because I've been complaining how expensive it is. And I said, we could probably do something different where you could look for between 1,000 to 5,000 loci that we could preferentially amplify and then sequence and use that as a much cheaper platform. Because once you made that upfront investment for 6,000 reactions, you basically have a panel that you can use for genotyping for a while if you're a small group like we are. And at that level, it would be about $1,000 per plate, right? Or 1,200 per plate. And then you could do as many as up to 6,000, I guess, assays altogether. And I said, okay, I'll explore it, but let's do this. Let's look at it. Let's evaluate it and see if it's actually robust enough for us. Because in our case, because we work with tetraploid roses, we want to make sure that we can get the dosages of the genotypes, in a robust manner. We already get it through the Axiom Array, so we just wanted to see whether it would be just as good as the Axiom Array, but doing a sequencing one and a lot cheaper too. And GCAN has an algorithm to get you as uniform as distribution of the markers as possible, so that in the end, the panel will represent most of the genome, will capture most of the genome anyway, and we could go with a service that would be more cost effective to the community. I don't think there's anything cheaper than that right now, and I hope that that's something we could do down the line. Thanks. Another alternative is Capture-Seq, but that requires a much larger investment to get that platform going. So this is the cheapest one we could do, because we would just base it on the Axiom Array and then just transform it into an amplification type, sequencing type platform instead. So we're in the middle of that right now, but it would be an alternative. A lot of folks are moving to DARTag, which is kind of like that. And Capture-Seq is another one. But again, this is what we can afford to develop right now, but it will be a useful platform to have if it works.